Hi everyone, welcome to our 7th session. Today I want to talk about the topic of irreducible complexity. This is one of the central tenets of intelligent design, and which I think many of you may have heard about before. But before I jump into talking about what it's about, I do want to qualify that this is actually a pretty controversial subject. There are people who feel like it goes too far in pushing for God's hand in evolution, pointing to many examples of complexity and saying, oh, because it's complex, God alone must have done this. Some see it as a God in the gaps kind of argument, where because we can't explain something, we just posit that God must have done it, rather than opening up the possibility for future scientific explanation. And while I do think that sometimes proponents of irreducible complexity can go a bit far, I also think there is merit in learning about it and knowing how to utilize it, because I think it's a useful way to open up the possibility that not all of nature comes from an undirected evolution. And so, irreducible complexity, how did it come about? This concept was made popular by someone named Michael Behe. He's a professor of biochemistry at Lehigh University in Pennsylvania, and it was in his book, Darwin's Black Box, that he first proposed this. As he's looking at the vast amounts of discoveries in biology, he noted that the deeper we look and we went into our understanding of the cell, the more complex things became. For example, when you open up the black box of a cell, it shows that life is based on machines and a complex system of regulation. And let's just watch a quick video to illustrate this. And so what we saw there are molecular machines hauling cargo from place to place in the cell. We see machines turning molecular switches on and off, sometimes killing the cell, at other times causing them to grow. Machines that regulate and facilitate replication, electrical currents carried along in nerves. In short, high levels of sophistication controlling every cellular process. The details of a life finely calibrated between all those different processes. And the question that Behe asked as he looked at all these things was this: could all of life be fit into Darwin's theory of evolution? Could all this complexity that he was noticing have evolved stepwise, undirected through multiple changes over time? Well, going back to Darwin's day, he looked at that, and the most complex organism at the time was the eye. By then, the anatomy of the eye was known in detail. The pupil acts as a shutter, the lens gathers light to focus on the retina, and this all changes depending on the colors and wavelengths to maintain focus. All these parts are necessary to function. And if you're just missing one of them, it would result in a severe loss of vision and blindness. And so the eye could only function if it were nearly intact, and it seemed to be an argument for design. Darwin himself said this. He said, if it could be demonstrated that any complex organ existed which could not have possibly been formed by numerous, successive, slight modifications, my theory would absolutely break down. At the time, it seemed that the gradual development of the eye was impossible because of many sophisticated features that seemed to be interdependent. But Darwin said that over generations, organisms could slowly accumulate beneficial changes in a gradual process. And they proceeded to show how this could happen in the eye. He proposed a series of steps to explain how the eye arose, pointing to modern animals with different kinds of eyes ranging from simple to complex, and suggested that these were the intermediaries for the human eye. And here's how he looked at it. He found that there are some tiny creatures who have a simple group of pigmented cells, a light-sensitive spot that can sense light and dark. Then, other animals such as starfish have a somewhat more sophisticated setup. Their eye is located in a depressed region. And since the curvature blocks off light from some directions, you can start to sense which direction light is coming from. And in fact, the more curved, the better directional sense, but also the lesser amount of light that enters in. And this sensitivity can then be improved by other organisms which have gelatinous material inside the cavity, which acts to focus the light a little bit more. Other animals start to develop a crude lens, and then gradual improvement in these lenses can gradually increase to more and more sharp images, leading to the eye that we have today. And when we look at these different steps, it looks pretty good. It sounds convincing that there's an evolutionary pathway from the simplest light-sensitive spot to the camera eye of a man. But the question is it really that simple? Well, think about the most basic aspect of vision, right? How does light go from a photon to something that the organism can perceive? Well, the mechanism wasn't known at the time. It was just assumed that it would be simple. You see light and then you react to it, and boom, you see. Darwin himself said how a nerve comes to be sensitive to light hardly concerns us more than how life itself originated. It wasn't until the more recent biochemical evolution that the steps involved were elucidated, and it turned out to be much, much more complex than anticipated. So I'm going to walk you through it. Don't worry about all the names, just appreciate the process. So this is what happens. When light comes and strikes the retina, it interacts with something called cis-retinol that then forces a change in the shape of another protein called rhodopsin and changes it to meta-rhodopsin 2. This then sticks to transducin and activates it. This complex binds to something called phosphodiesterase, PDE, on the inner membrane. Activated PDE then cleaves cGMP, cyclic GMP, which then closes the sodium ion channel, decreases the cellular concentration of sodium, and triggers the optic nerve. And that is interpreted as vision. That's what it takes. And then via other mechanisms, it resets the system so it could be ready to receive the next photon. I mean, it's pretty amazing, isn't it? And this is what it takes for vision to happen at the molecular level. And so when we talk about evolution with modifications, we can't just talk at the higher anatomical level, because you can easily say, well, this intermediate went to that one and to that, and we have an explanation. No, but the question is, what does it take to go from intermediate to intermediate? And it's this level of sophistication. To do what Darwin did and just look at the intermediates and hypothesize they just transition from one to the other, that's not enough. It could be, but we have to talk about how, especially since evolution talks about changes due to a series of numerous successive slight modifications. And it's at this inner biochemical level that these changes are actually made. Now, as you think about this description of the eye, it's as if you were to look at a motorcycle and wonder, how does a motorcycle get made? And then you look at a bicycle and you're like, it must have gone from a bike to a motorcycle. After all, they look similar. They have two wheels, the handlebars, light, seat, etc. And over lots of times with a series of small successive changes, it could happen, right? Well, maybe if a teenager spent a lot of days taking an old lawnmower engine, other spare parts, they could. But to have a series of gradual successive changes by chance without some sort of directed intelligence? If you start to get into the details of it, the quote biochemistry, you realize that it really could not have happened. A motorcycle requires fuel. The bike has nothing that can be slightly modified to become a gasoline tank. And then what part can be duplicated or modified to build a motor? And even if you were to get a motor by chance, how can it then be mounted and connected correctly to the drive chain and to motorize a bike? How could this be done step by step with small modifications in an undirected way? In fact, when you start to look at the details, it's much more complex than you thought. And you start to cast doubt that a motorcycle can come with a bike with a series of successive modifications. And in fact, there's been no example in history that has shown this. And so what advances in biochemistry did is that it opened up the black box of the cell, unveiled the complexity beneath it, and seeing that to make these outer changes step by step was not a simple matter that we could so easily attribute to evolution. This level of biochemical complexity provided a hurdle for evolutionary biologists to explain. And so that's the complexity piece of irreducible complexity. But what about the irreducible? Again, going back to what Darwin said about complex organs. He said, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Well, what other type of biological system could not be formed by numerous successive slight modifications? It's a system that is irreducibly complex. And so what does that mean? Well, a system that's irreducibly complex means a single system composed of several well-matched interacting parts that contribute all together to the basic function, where the removal of any one of these parts causes the system to effectively cease functioning. Okay, and when you look at a system like that, such a system cannot be produced by slight successive modifications, because any precursor that is missing all of a sudden makes the whole thing non-functional. The whole thing falls apart. And since natural selection only chooses systems that are already working, what happens? Even a partially constructed one, built halfway, that's still not functioning yet, and it would not be selected for. In fact, all the parts have to arise as a single integrated unit in one fell swoop in order for natural selection to have anything to act upon. And so the question is then, when can you be sure that something is an irreducibly complex system? Because there are systems out there that are complex, and yet when you remove certain parts, they function just fine. But then there are other systems where if you were to eliminate even one part, the whole system would fall apart. And so here's Michael Behe to explain this a little bit more.
You and I are made of about 40 trillion cells. They comprise every part of our complex human anatomy. But the truth is that we can reduce the number of parts and still have function. If we lost an eye, an ear, or a hand, it would be very unfortunate, but our lives could go on. The same applies to this rather complex drill. It has some cool add-ons, but I could take off this battery gauge, or this LED light, or a few other gadgets, and I'd still have a working drill. But how far can I reduce its complexity and not ruin it? I don't want to take apart my drill, so let me move to another example. This mousetrap. The complexity of this machine entails 5 parts: the platform, the spring, the hammer, the holding bar, and the catch. Now, if I reduce the number of parts from 5 to 4, if I take away the holding bar, or the spring, or any other part, it wouldn't work. Every part is essential. I call this irreducible complexity.
All right, so I want to break down irreducible complexity a little bit. So how do you determine if something's irreducibly complex or not? Well, the first step is to specify both the function of the system, like what is the whole system doing, and then its components. And what you want to find is something that's composed of several parts that all contribute to the function. And here I want to look at the classic model that Behe proposed of the mousetrap. The function of a mousetrap is to immobilize a mouse, so it won't keep chewing through food or furniture. Like at Potomac House here on the East Coast, we recently had mice problems, so we put out all these mousetraps. And so what does a mousetrap consist of? It consists of a number of parts. And what are the parts? Well, there's this flat wooden platform that acts as a base. There's a metal hammer which does a job of coming down on the mouse. There's a spring. There's a sensitive catch that releases when some sort of slight pressure is applied. And then there's a metal bar that connects the catch and holds the hammer back until the trap is charged. Now, once you outline the parts that make up the system, you then need to ask, are all of these components required for their function? And so in the case of the mousetrap, they are. Right, so imagine one of these parts are missing. Would you be able to catch a mouse? Well, say the wooden base were gone. Then there would be no platform for attaching to the other components. Well, what if the hammer were gone? Then there's no way to pin down the mouse to the wooden base. There's no spring, the hammer and platform would just dangle and not be able to perform its function. And if there's no catch or if there's no holding bar, then the spring would just snap shut as soon as you let go of it. And so you would say that this mousetrap is irreducibly complex. It's made up of interacting parts that contribute to its basic function, and removal of any one of these parts causes the entire system to stop functioning. And so it makes sense that this thing would have to have been made all at once in a way that's directed towards some sort of end product. Right? This idea of directionality, directed, working towards a certain end product, these are all features of an intelligent rather than an undirected process. And so that the natural question to ask is this: are there examples of irreducible complexity in biological systems? Systems that cannot be explained just by Darwinian changes. To identify these, we need to find systems that have multiple parts that contribute to its function, and then be able to specify their essential roles, without which the whole thing would fall apart. And so when we think about the idea of irreducible complexity, the most famous example is that of the bacterial flagella. The flagella is this corkscrew-shaped hair-like appendage that's attached to the cell surface, acting like a propeller so the bacteria can swim. So here's Behe again to explain this.
Here's a bacterium, a single-cell microbe. Some bacteria have a flagellum, which is like an outboard motor on a boat. It rotates to propel bacteria through liquid, and it's necessary for their survival in order to swim to search for food. A flagellum has a number of parts: a drive shaft, a universal joint, a rotor, bushings, stator, even a clutch and braking system. The motor of the flagellum has been clocked at 100,000 revolutions per minute. And as with the mousetrap, removing even one component of this elegant machine destroys its function and renders the bacterium, shall we say, dead in the water. The flagellum is irreducibly complex.
The flagella's motor is much like an outboard motor. It's a single protein whip that's attached to a tiny electrical motor made up of different kinds of proteins, as you see here. It can be essentially broken down to 3 main components. First, a paddle, a flagella, and then a rotor, some sort of rotating element, and then a stator, a stationary element that helps keep it there while it spins. Each of these components are essential for function. If one were missing, the flagella is unable to corkscrew and do its job of propelling the bacteria. As with the mousetrap, it's difficult to see how Darwinian's gradualistic process of natural selection with random mutations could produce this because there's all these pieces that are required for the function to appear. A flagella by itself or a rotor by itself would not act as a propulsive device. You need all three of these parts working in concert. And scientists have looked at the flagella and they've acknowledged that actually it does appear like it's designed, as it's complex and functions much like motors. But the question they ask is it really irreducible? Could it not have been built from a series of parts that were used for other purposes and now are put together? For example, what if the tail had some other function, like to detect the flow of fluids and instruct the bacteria in which direction to go? What if the motor, which looks kind of like a pump, had a function of protecting the bacteria? And then through random changes over time, these came together to form an evolutionary advantageous flagella. That would be an indication that even though it appears irreducibly complex, it actually is not. And scientists have claimed to have found one such intermediary. And what that is something called a type 3 secretory system, T3SS for short, which I'm going to call it from here on out. And it's found in a subset of bacteria. It's a pump that's used to transport proteins across cell membranes. In particular, it's used by disease-causing bacteria to punch holes in the host target cells. The structure actually looks surprisingly similar to the flagellar system, and in fact shares almost identical proteins to it. Based on this, scientists have hypothesized that evolution borrowed the T3SS, repurposed it, added on a bunch of different components to then get the flagella. And therefore, the flagella is not irreducibly complex. Or is it? And so it's true that 10 of the proteins in the flagella are very similar to that of T3SS. What about the other 30 proteins that are there in the flagella to make it work? The question is, where are you going to borrow that from? And what kinds of function did they have before? More than this, how is it that these parts work together to transform the T3SS from its initial function of just secreting proteins to now this amazing molecular machine? Even if all the parts are put together in the same place, how do they get assembled in a way to make the flagella function? You can't just put a few of them together. All of them have to be put together in the right way in order for the flagella to work. And how do small, gradual changes account for that? Well, we can analogize this dilemma to the mousetrap. It's something like this. Like you think about a mousetrap, how did it form from a series of parts? What if the board was like a popsicle stick? And the spring was something that you got from an old clock? The metal hammer was like a crowbar, one of these fish-high locks for the hold bar, and then a bottle cap as a catch. Each of these had some sort of different function before that they had been selected for, they were functioning, and now you could put them together and perhaps that could work. But what if we start to put these together? Are we even close to a mousetrap? No, they couldn't even form a functioning mousetrap without extensive modification and assembly. And then what steps would it take to get there? It's very difficult to imagine through random chance and selection. It requires some sort of intelligence to direct it towards some sort of end, towards a function that you're aiming for. And then here's the other thing. While modifications are going on, what happens? These original things, like a bottle cap, wouldn't be well suited for their prior role. Nor would they be suited for their anticipated function as a mousetrap. And so if they're not functioning well for those things, they would quickly be selected out. So in fact, the presence of the secretory system does not serve to diminish the irreducible complexity of the system. Dembski himself said this. He said, finding a subsystem of a functional system that performs some other function is hardly an argument for the original system evolving from that other system. One might just as well say that because the motor of a motorcycle can be used as a blender, therefore the blender motor evolved into the motorcycle. Perhaps, but not without intelligent design. Indeed, multi-part, tightly integrated functional systems almost invariably contain multi-part subsystems that serve some different function. At best, the T3SS represents one possible step in the indirect Darwinian evolution of the bacterial flagellum. But that still wouldn't constitute a solution to the evolution of the bacterial flagellum. What's needed is a complete evolutionary path, not merely a possible oasis along the way. To claim otherwise is like saying you can travel by foot from LA to Tokyo because you've discovered the Hawaiian Islands. Evolutionary biology needs to do better than that. And then here's another way to look at it. Since the T3SS and flagellar system look similar, there are 4 different possibilities to explain their relationship. First of all, T3SS is the ancestral form from which the flagellar system evolved. So in a way, it was co-opted by the flagellum to use to create the more complex flagellum that we see today. Option 2 is that the flagella and T3SS evolved from a common ancestor that was more primitive than both of them. Option 3 is that the flagellar motor is actually the ancestral form from which the T3SS is degenerative byproduct. In other words, the flagellar motor was present first, but then a more simplified, quote, degenerative version was developed in the T3SS. And then the final possibility is that both of these are just completely independent in their origins. The first two of these hypotheses actually contradict Behe's hypothesis. The second two support it. And so these different possibilities give us different testable predictions. For example, number one, if the co-option hypothesis is right, then the T3SS should have genes that are older than the ones that built the flagella. But actually, if you do research, you find that research has shown that those genes that built the T3SS are actually younger than the flagellar ones, suggesting that the flagellar is actually the more ancient one, and the T3SS is a derived one. And here are some lines of evidence that show that. First of all is something called mutation density studies. And what they do here is they look at the mutational density in a different set of genes. They look at the genes and they say, what's the degree of variations that we see? If there's a lot of variations in that gene, they assume that that must mean that there has been a lot of mutations, which means a lot of time must have been elapsed since this gene was established. If there's less variation, then less time has elapsed. And what they find is that the mutation density in the T3SS is less than that of the flagella, and that implies that the flagella itself is older. The T3SS actually developed later or independently. The second line of evidence is called phylogenetic distribution. Here's a typical phylogenetic tree, and it shows us how species develop. And what you'll imagine is this, that older structures will be present in more organisms because they arose later on, in the earlier past. Whereas those things that arise later are only found in a small subset, right? Only a small subset of them will show this structure. And what we find is when we look at all the different species of bacteria, the T3SS is only found in a small subset of bacteria, kind of later on in evolution. Not in gram-positive ones and not in a majority of gram-negative ones. It suggests that it hasn't been around that long. But the flagella is found and widely distributed across all groups of bacteria. It suggests an origin deeper in the phylogenetic tree, in other words, older. And the third thing is something called the age of eukaryotic cells. So we look at T3SS, these are used by bacteria to inject toxins into what? Eukaryotic organisms. These are organisms with a nucleus to digest their contents. When we look evolutionarily, eukaryotes actually come much later in life. And so when you think about function and the selective pressure, you would need a flagellar motor much earlier in evolutionary history than toxin production for these eukaryotes, which come much later in life. And so the flagella conveys a much more basic function. These evidences count against hypothesis number one. And if you think about them in hypothesis number two, if they're co-derived, you expect genes to be of the same age. But as we saw just now, there's a clear temporal asymmetry that the flagella do appear much older than the T3SS. So option 2 doesn't really fit as well. And so looking at this, Howard Ackerman, a professor of molecular and evolutionary biology, said, the most parsimonious explanation is that the T3SS arose later. The T3SS is likely a broken flagella, which is quite possible given that in Darwinian evolution, most mutations are the loss of function. And so it shows that this supports Behe. And so the flagella still stands as an example of irreducible complexity, which cannot be explained just by a series of small successive changes. But irreducible complexity is not just confined to the world of prokaryotes and bacteria. We see it in many processes that even our body uses. In fact, it's pretty amazing. Part of the reason I wanted to become a doctor in the first place. When we talk about irreducible complexity, we usually talk about something called the blood clotting cascade. But for today, I thought it'd be fun to look at the immune system. With COVID and all that, it's a way that we can appreciate the body's defense mechanism as well as a few varied examples of irreducible complexity. It's going to be pretty detailed because in order to appreciate irreducible complexity, we need to know the complexity of the necessary components. So I'm going to have you channel your AP Bio self, and here we go. So when a pathogen enters our system, the immune system automatically gets triggered in a way that serves to protect our body. And as we look at how it does it, we see that it's made up of coordinated processes that are each irreducibly complex. And the two I'm going to talk about today are something called antibody diversity and the complement system. So, antibody diversity. If you think about it, when we encounter a novel pathogen just like COVID-19, which no one on earth has ever seen before, how does your body fight it off? Why doesn't everyone succumb to the virus? How does your body produce the right antibodies necessary to fight off COVID-19 infection and recover? Well, here's a really interesting experiment. They took these rabbits, and what they did is they injected synthetic molecules into them. These are molecules that are completely novel. They're never found in nature. No rabbit has ever seen before. And yet what they found is that these rabbits produced antibodies to these molecules. And the question was, how did they do that? They've never seen them before. And so one hypothesis was, did these proteins flexibly wrap around these antigens and somehow freeze their configuration? Or are there vast numbers of genes for antibodies all throughout with different shapes to allow for recognition of these random invaders? But if you think about it, that would mean billions and billions of combination. That would take up more space than there are coding regions in DNA. So how does the body do it? It's crazy. Well, the answer came in the form of something called splicing. Scientists discovered that genes were transcribed from the DNA and they had these things called introns and exons. But the coding segment is just the exons. And so what they found is RNA actually edits out to get the right message in a process called splicing. And they looked at it and they found that actually antibodies are the same. When they look at DNA, there's splicing, rearrangement of the DNA in each antibody producing cell. And what it does, it starts to give diversity to the antibodies that it carries. And so let's dive in a little bit more to see how this works. So for antibody formation, there are actually 4 gene clusters in these antibody producing cells. There's 40 genes in this V region, 23 genes in the D region, 6 genes in the J region, and 3 genes in the C region. And what the body then does, what the cell then does, it takes one segment each from the V region, the D region, the J region, puts them together with something called a VDJ recombinase. And how does it do that? Well, there are these special DNA sequences that flank each gene segment, and it serves as recognition sites for joining and ensuring that only the appropriate segments recombine. And this is the process for forming something called the heavy chain part of the antibody. But actually, a very similar process goes into forming the light chain. And what happens is when you calculate all the different combinations that can arise from this, you get this astounding number that there are 10 billion combinations of antibodies you can form just from these few hundred segments. And then there's a whole other process through which once there's a pretty good match, the antibodies start, the cell gets selected out, it starts to hypermutate, so you get an even better fit. It's a pretty amazing system. And I remember when I first learned about this in med school, I was astounded. Such an ingenious way to create diversity to recognize whether an invader comes to the system. And then what you also notice, as you really think about it, is that it has characteristics of being irreducibly complex. How? Well, let's go back to this. What are the components that are required for antibody diversity? Well, it's the genes themselves. It's also the signals that identify the beginning and end of segments, telling where they're joined together. And then there's the machinery, the VDJ recombinase, that recognizes these cutting signals and joins them together in the right order. And so if you think about these different components, without the machine, the parts never get cut out and joined. Without the signals, the machine just randomly cuts. You don't get the antibodies you need. And without genes, of course, it's pointless. All these things have to develop together. And here's the thing, even if you manage to get all these pieces together, they'd have to function pretty efficiently from the start. Imagine a primitive system where only a few antibodies were formed. That would be like an FBI database with only a few fingerprints. The probability is so small for these antibodies to be complementary to an invader. It would be too inefficient to actually be selected for, right, in a process that's undirected. To do any good, you have to generate a lot of antibodies from the start. And here's the thing. This is not the only irreducibly complex part of our immune system. I'm going to go over the next part called the complement system because it illustrates a different type of irreducible complexity. Again, the names are not so important as much as the concept, but we need to describe the processes so you have to appreciate the complexity involved. And so here it is. This is the complement system. So when you think about antibodies, they actually don't harm anything. They only stick to the invader. They look kind of nice, like stickers. The killing is done by something called the complement system, which binds these antibodies and activates the destruction of the cell. So how does this work? Well, there are two main aspects of the complement system. I know it looks complicated, but just bear with me. There's something called the classical pathway and an alternative pathway. Those are the two main ones. What happens in the classical pathway is a protein called C1 binds that bound antibody. And then once it does, it changes shape and it activates the rest of the cascade by splitting something called C4 and C2. These then combine with C3 to form an activation unit. The entire complex then cleaves something called C5. This activated C5 then sticks to C6, 7, 8, and 9, and it starts to form this unit called the membrane attacks complex, which then punches holes in the membranes of the cells, water rushes in, and the cell bursts. The cell dies. That's how it defends itself. There's also another pathway called the alternative pathway that doesn't need to wait around for the antibodies. A small amount of C3b is continually produced in small amounts. This gets destroyed unless bound to a membrane upon which it binds to something called factor B. And then when there's more C3b, this then forms into something called the C5 convertase, which then goes around the same system to lead to pore formation. Yeah, I know. Really, really complex. Everything interacting with each other, dependent on one another. And that's just a surface level. That's why med school is hard. But in terms of explaining the pathway via evolution, right? How do we explain this by step-by-step processes? Well, creating the pore, that's not hard, right? You just have one killer protein, you can do it, that's fine. The issue with Darwinian evolution explaining this is the control system, because it's a series of switches that are dependent on one another. At each point, the regulatory protein, the mass protein that activates, they have to be present from the beginning. Because you miss one of them, what happens? Right? Take C5, for example. If C5 is activated, if it's C5b in the beginning, well, boom, the rest of the cascade just goes off and everything starts lysing. You don't want that to happen because you'll lyse your own cells. You'll get rid of your own cells. But if C5 is just there by itself with nothing before it to activate it, then the whole pathway is off. The prior steps have to be there. Then what about the switch before it, C3? Well, if the activated C3, C3b is present, boom, the cascade goes off. But if just C3 is there, the whole pathway is off. And so you have to get activated by something else, and you've got to go back more in time. Well, what about the binding of C1? Well, if the binding of C1 to the antibody did not trigger activation, then there's no pathway. But if the activated C1 is present before binding the antibody, it would be prematurely triggered, and again, all these cells would start dying. So in other words, all these pieces have to be in place, ready for the system to activate in order to be regulated and work. When you miss one of these systems, either it doesn't work, or if it's activated, it'll be premature. They're all dependent on one another. It's kind of like this machine. It's called a Rube Goldberg machine, composed of several interacting parts that contribute to its function. All these things in order to staple a paper. It's kind of fun to see. But removal of any part here would cause it not to work. So unlike the prior examples of irreducible complexity, like the flagella and antibody production, it's not a single complex unit made up of different parts. Rather, it's composed of separate pieces, each acting in turn, one upon another, to accomplish its function. And so the entire system itself is irreducibly complex. All the parts need to be in place for the entire cascade to be appropriately regulated and function. Like, how did it come to stepwise fashion? It can't. It all had to have been there, ducks lined up in a row, so that you can kick it off and all the things happen. And so what we see here with antibody diversity and with the complement system is that each of these by itself is irreducibly complex in different ways. And on top of that, what's really amazing is these interact with one another, and these are actually dependent upon one another. And so, for example, a large antibody diversity is no good without a system to kill the invaders. And the system to kill the invaders is no good without a system of identification first. And so each of these is interdependent on one another as well. The immune system is incredibly complex, each part dependent upon one another, each process working in concert with which the whole thing, if it didn't, would fall apart. And so even the whole process itself is irreducibly complex. And there are other systems like this, and we're discovering more and more about how our body works. So the question is, how do we explain these findings of irreducible complexity in nature? Evolution itself demands that things appear by a series of small successive changes. That's how it works. That's the only mechanism it works. But that alone doesn't explain these irreducible complex systems that we're seeing. And so are there other theories to account for that are undirected? If not through a series of small successive changes, then what? Perhaps they could be put together quickly or suddenly. Or if adding individual pieces doesn't work, then what if you add multiple pieces together? Are there mechanisms that can do that? Well, here's one of them. It's proposed by a scientist called Lynn Margulis, and she kind of titled it Advancement by Cooperation Symbiosis. Big words, big names. But basically what it's saying is that organisms aid one another. They join forces, symbiosis, and accomplish together what they could not have done separately. And this came about because when you looked at cells, they're full of complex machines that are separated into discrete compartments. The biggest compartment is nucleus, the smallest one is the mitochondria. And when she looked at these things, she proposed that actually parts of these cells were once free living organisms. She looked at the mitochondria, for example, and she found that it was roughly the same size and shape as a free living bacteria. And she proposed that actually this is what happened, that there was this larger cell that then swallowed up these smaller bacteria. But rather than digest it, they start to work together. The smaller bacteria receive the nutrients from the larger one and then pass on energy to the larger one. They then start to reproduce together, and over time they specialize more and more. And now the smaller cell became the mitochondria. And this is true also of other compartments as well. This theory became much more accepted over time when they sequenced and they showed that the mitochondrial proteins more closely resembled the bacterial proteins than the host cell itself. And they looked at this and say, wow, this is how you can have complexity arise without small successive changes. Boom! All of a sudden, all these new functions come. But the question is, does this explain the irreducibly complex biochemical systems we've been talking about? Well, actually, it does not. Because what we see in this theory is that it's the symbiosis of two cells, each of which is functioning already. And the question is, how did these pre-existing cells originate in the first place? What's the origin of their biochemical irreducible complexity present in each one of them? And the theory is unable to answer those questions. So what does? And here we want to turn back to what we talked about when we talked about the origin of life. What makes the most sense with these irreducibly complex systems? Isn't it plausible to consider some element of design? After all, what is design? Design is a purposeful arrangement of parts. Like when my kids take a bunch of Lego pieces and purposely arrange them to form this cool spaceship. Isn't that what we're kind of seeing here with irreducible complexity? Some sort of direction, purposeful arrangement. If you think about it, how is it that we detect design? Remember, we talked about specified complexity. And what is that? That's low probability events that have a specific pattern that indicate a function. For example, the flower arrangement that you see spying on a message. The pattern of signals from outer space that signal what SETI is looking for, or Mount Rushmore. Seeing things ordered in a way that's directed towards a purpose. And in an analogous way, as we look at different irreducibly complex systems, aren't these the same? They're low probability events whose pieces seem arranged in a way that all the parts need to be in the right place in order for the function to happen. Miss one, and the whole function falls apart, like what we saw with the complement system or with the Rube Goldberg machine. Or what about this? When we look at the complexity systems like the flagella or the immune system, it doesn't appear to be a gradual route through which the individual components could be assembled in an undirected fashion. But when we look at things like that in daily life, something like a motorcycle, we assume design because the parts fit in a way that must have been directed towards a specific function, put there for a specific purpose. And we can infer that that's not only true for designed inanimate objects, but as we look at these complex biochemical systems, we can say, yes, it actually has the appearance of design. In fact, that's a possible and a plausible explanation. Now, some people argue that irreducible complexity is a, quote, God in the gaps argument. And what it is it accuses of inferring God just because it can't explain how things came about. That's actually not what this is. We're not talking about a gap in knowledge here. The gap we're seeing is in the Darwin's evolution difficulty in explaining how we can get complexity through a step-by-step, undirected process. Because in these systems, if you miss one part, the whole thing falls apart. Or you need all the pieces together in order to get a selectable function. And these scenarios, step-by-step, won't cut it. And so what is the other explanation? Design is a perfectly plausible explanation. And so that's it for today. We'll be going deeper into intelligent design. And Richard's going to be talking for the next 2 weeks about evolution. So I'm really excited for that. I'll see you guys later.